Okay, let's get started. So it's my pleasure to introduce Andrea Putnam, who's a brand new PI in biomolecular chemistry. Andrea got her PhD with Eckhart Jankowski, working on helicases, and then went to Geraldine Seydoux' lab at Johns Hopkins to work on maternal RNAs and stuff, probably some stuff we're going to hear about. You know, when I was a graduate student, organelles had rules. They were simple. They occasionally let some things in. There were like gatekeepers and all sorts of, occasionally let some stuff out. And then all hell broke loose. And this was obviously before there were fluorescent markers. This was before high resolution microscopy. And so now this, what was originally a developmental biology problem, maternal RNAs, really is a crossroads of genetics and cell biology, and has direct applications to all sorts of things, neuroscience, the field that I'm in, and pretty much everything else. So I'm excited to hear about this, and here's Andrea.
Okay, can you all hear me today? Great. Okay, so thanks so much to Jerry for inviting me and for the introduction. And I'm very excited to talk to you today about some of the work that I did as a postdoc and then what we're doing in my lab now. And I'm excited to present what one of my first graduate students has been working on. Today I am going to talk to you about maternal mRNA localization in C. elegans embryos. But I'm also going to tell you about the structures that we think are important for localizing these RNAs, which are these membraneless organelles, as Jerry was introducing, that are actually pretty complicated. And in our lab, we're interested in studying these structures both in animals using C. elegans as a model, but then also trying to reconstitute them in vitro to really understand both the biological and the biophysical and biochemical properties of these structures. So in my talk today, I'm actually going to start out with a little bit more of the biophysical properties of these granules and kind of what we think they're doing in worms, physically and structurally. And then for the second half of my talk, it's going to be focused on the role that we think they're playing in maternal mRNA localization. Okay, so yes, so we think about how cells are organized a lot in the lab. Oh, and okay, there we go. So we think about how cells are organized, and canonically, right, we thought about this as membrane-bound compartments, nucleolus, mitochondria. But really, in the past few decades, there's been an explosion in understanding that cells are also compartmentalized into membraneless structures. And there have been many of them that have been identified recently. Some of them we've actually known about for more than 100 years, which is the nucleolus, which is a membraneless organelle found in the nucleolus of almost all eukaryotic cells, and it's involved in ribosome biogenesis. Then there are other condensates that you can find in almost all animals, but only form under specific conditions. For example, stress granules, which form when there's environmental stresses. In the lab, we're really interested in germ granules. And so germ granules are a type of germ granule that only form in specific cell types. In our case, this is in germ cells. And so we're really interested in germ granules for several reasons. First, they're found across metazoa, so from C. elegans, zebrafish to humans. We know that when germ granule components are disrupted, it can result in defects in RNA processing and cause sterility or infertility. And so there are a lot of germ granules are actually complicated. So we call them as a class. Any membraneless organelle that's specific to a germ cell is a germ granule. And so there are actually multiple granules, but some of them have components that are very highly conserved, such as this protein, which is shown here in red in C. elegans and zebrafish, and green in human spermatocytes. And so this is the marker for an RNA helicase, which is GLH1, VASA, or DDX4, and you can find it in germ granules throughout evolution. So despite the fact that these structures are conserved and probably doing something important, there's a lot that we don't know about their biology. And so we've been using C. elegans as a model to try to understand what these structures are doing in development. And so C. elegans has been a really powerful model for studying membraneless organelles. And they were first identified in 1983 by Susan Strome, who found an antibody that identified these really bright puncta in germ cells throughout the life cycle of the worm. So from the unfertilized oocyte into embryos and larvae and into the adult germline. And the protein that she identified was this protein PEGL, which is shown here with a fluorescent GFP marker. And this is a one-cell embryo. So you can find PEGL in granules throughout the life cycle of the worm, but they undergo some really dramatic changes. And PEGL is just a marker. There are a lot of other proteins and RNAs, and in fact, other germ granules that change a lot as you go through development. So I just kind of want to point out now, this is not really one structure. And in my lab, we're mostly interested in studying these granules in the early embryo. And so in early embryos, these granules are cytoplasmic. When you have in an oocyte, you can find granules throughout the cytoplasm of the oocyte. But after fertilization, when this cell divides, granules become polarized. And I'm going to show you that in this movie here in just a second. Later on during embryogenesis, so this cell will undergo several asymmetric divisions, and you'll have germ granules that are left in one cell, which will go on to become the germline founder cell. And in larvae, these cells will then proliferate, giving rise to the two gonad arms of the adult worm. Okay, so as I mentioned, we're really interested in early embryos. So after fertilization, you have an embryo. P granules start localized throughout the cytoplasm. As the embryo goes through mitosis, these granules dissolve in the anterior half of the embryo, and they'll grow in the posterior half of the embryo. And this whole process occurs in about 10 minutes. And so I'm going to show you this here. Nuclei are marked with a histone marker in red, which is a little hard to see, but you can kind of look for as the movie goes through. Right, so what you'll see is these granules dissolve in the anterior, and they grow in the posterior. Our cell is just about to complete mitosis here, and we have now 2 new nuclei, and we now have a 2-cell embryo. And the germ granules are only in the posterior side of the embryo. Okay, so as you can see, these are very dynamic structures. They're dissolving and growing. And they were actually one of the first intracellular granules that was proposed to form through a process called liquid phase separation. And so this is a process similar to, for example, if you mix oil and water. Oil is not very soluble in water. It will phase separate and make liquid droplets suspended in here water as a solvent. This is very similar to what's going on in this cell. You have an emulsion of droplets that are formed from RNA and protein, and they're suspended in your dilute phase, or the cytoplasm in this case. What's very cool is we can take individual proteins and even some RNAs from these granules, and they will make droplets in vitro. So we can study them not only in cells, but in a test tube. And when we look at these in vitro, you can see these dynamic liquid properties where you have droplets that can fuse and grow over time. Okay. So this mechanism was proposed by Cliff Brangwyn in 2009 using P granules as a model. And it's really been a powerful model for thinking about how membraneless organelles form and what their properties might be. So I want to just take a couple minutes to actually walk through why we think this is such a cool model for cellular organization. Okay, so first, phase separation is a spontaneous, thermodynamically favorable process. So once a component exceeds a certain concentration and it's no longer soluble in whatever your solvent is, cytoplasm, for example, When you add more of your protein, instead of dissolving, it will form droplets. And as you increase the concentration of protein, you'll just get more droplets. And the concentration of protein that's in the dilute phase will stay the same. Okay, and so again, this is just a thermodynamic process. That means it doesn't need energy. So if you're a cell, you just need to have a component that gets to a high enough concentration and you can compartmentalize it. This also right away gives you a potential cool function of these droplets, because as you change concentrations, the concentration in this soluble phase is going to stay constant. The concentration in the droplet, you're going to get more bigger droplets, but it's also going to be constant. And so this has been proposed as a way you can buffer changes in cellular concentrations. And so this is one of the ideas that may be a function for condensates. Condensates also contain mobile components that can exchange with the dilute phase. So this is different than a membrane-bound compartment, where it usually takes energy to transport molecules across membranes. Here, proteins can exchange from in their concentrated environment in the droplet into the dilute phase. As you might imagine, how things are soluble is really dependent on the environment. So where this phase boundary forms is going to be sensitive to changes in environment, including temperature, osmolarity, things that change, for example, protein properties like post-translational modifications. Then finally, the last feature I want to point out of liquid droplets is they're spherical. And this is not actually trivial. It tells you something about the underlying biophysics of their assembly. So as you might imagine in a droplet, the molecules at the surface of the droplet are going to be interacting with the solvent more than molecules in the interior. And so these are going to be less energetically favorable. So there's a drive to reduce the number of molecules at the surface of these compartments. And a sphere is the shape that's going to give you your lowest surface area to volume ratio. And so you can see this here. I'm going to play just a quick movie where you have two droplets. They collide and fuse, right? And they're going to rapidly relax back into a sphere. And this is driven by surface tension. Okay. All right. So one more important thing I want to point out about liquid droplets is that when you see many liquid droplets suspended in a solution, this is also called an emulsion. This is actually unstable, right? You have a lot of small droplets with a lot of surface area. They want to minimize surface area over time. And so you should get coarsening, where you have small droplets grow into larger droplets until at equilibrium, you're only going to have two phases, a dilute phase and a condensed phase. And this is similar to, for example, if you have a vinaigrette, you shake it up, you make an emulsion. Over time, those will separate out into two phases, an oil and an aqueous phase. And we can see this in our in vitro emulsions, the video I showed you before. Here you have a bunch of small droplets. They're coalescing or coarsening, resulting in many fewer droplets. So it's approaching equilibrium. But in cells, we actually rarely see this. We usually see many droplets. So it's worth thinking about when you think about condensates in cells, is they're probably not at equilibrium, or there are going to be processes in the cell that are preventing this from occurring. Okay, all right, so that's kind of like your basic primer into liquid droplets, some of their properties, and how we think about them. The other thing that I want to tell you is that droplets, especially in cells, are complicated. And so this is known in material science and food science for a long time, but I think something that as biologists we're really beginning to appreciate. So if you mix different components that form liquid droplets, they don't all mix together. Instead, they can make their own individual compartments that can make these kind of cool multi-layered structures. So here, this is just shown in an image from the Brangwyn lab, where they took water and oils labeled with different dyes, and you can get this kind of cool arrangement of multiple compartments. And this has been seen in most cellular condensates. So this is a little bit trickier in cells. Cellular condensates are usually less than a micrometer in size. So a lot of things look kind of like a big droplet when you look at them with regular microscopy. But as we're looking at more of these with improved microscopy and super resolution techniques, it's clear that most of these condensates are more like this, where you have many different substructures, including small condensates and a bigger condensate, or even things, you can have something that looks like a bunch of different types of liquid droplets within other liquid droplets, but then also things that are more complicated, where you may have fibers in a liquid droplet. So this is kind of here just to say, in biology, it's complicated. So we're thinking about these as liquid droplets, but it's not necessarily as simple as just one droplet. And this is something that we really care about in C. elegans embryos, because we found that germ granules in embryos are complicated too. And so here we're looking at an embryo. It has just polarized. So you have a 2-cell embryo, you have P granules in the posterior and the anterior. This is the protein PEGL, which is one of our kind of a core marker of P granules. And in vivo and in vitro, it behaves like a liquid droplet. And even at high resolution, it makes these nice round spherical structures in vivo. But we've been working on other proteins in granules, and particularly we're interested in this protein MEG3, which is really important for regulation of granules in the early embryo. And when we look at this protein with high resolution microscopy, we can see that it doesn't mix with the PEGL phase. Instead, it forms small clusters on the PEGL condensate that are actually on the surface of this condensate. And so these little surface clusters decorate the surface of the granule. Through work I'm not going to have time to share with you today, we've confirmed that this PEGL core really behaves like a liquid, but these MEG3 clusters are actually solid. They're little nanoscale solids, and they're not dynamic. Okay. So as a postdoc, a lot of my work was really trying to understand what this protein MEG3 was doing, and if its organization, if this structure was important for what it was doing. And so, as I mentioned, the first part of my talk is going to focus on just showing why we think these structures are maybe important for how MEG3 regulates condensates. And then the second half of the talk will be focused on the role we think that MEG3 is actually playing in the localization of mRNAs in embryos. Okay, so as I mentioned, in our lab, we like to purify proteins and see if we can reconstitute things. So one of the first things that I worked on as a postdoc was trying to reconstitute this organization in vitro. And so we purify proteins in bacteria, the C. elegans proteins. We can label them with fluorescent dyes, so we can look at them on a microscope. And we try to purify them in conditions where they're soluble, so they don't make granules. And then usually that's going to be something like high salt. And then we can dilute them into a buffer that has low salt and approximately physiological pH, and they will make droplets. And so we can do this with PEGL3. So if you take PEGL3, put it in a test tube, it'll make these nice big round droplets that will coarsen over time. If you take MEG3, it also makes clusters, but unlike PEGL, these little clusters that form, they don't get bigger, they don't grow over time. They're just little solid particles that kind of hang out. And so what's cool for us is we can take these two solutions, our PEGL liquid droplets, add MEG3 to them, and MEG3 will actually decorate the surface of these droplets in vitro. And we look at this at higher resolution, we can see that these MEG3 clusters decorate the surface of the granule, very similar to what we saw in our in vivo in embryos. Okay, so when you think about a particle that's on the surface of the condensate, I spent a little bit of time talking about surface tension and how that drove the shape of droplets. So if you see an organization like this, your first thought should be maybe MEG3 is doing something to the surface tension of these droplets, because it's at this interface between your droplet and the solvent. And in fact, this is a mechanism that's been described in the physical sciences, so in material science or in food for a long time, where if you have liquid droplets that can coarsen over time, and you just add nanoscale solid particles to them, they'll coat the surface, reduce surface tension, and prevent coarsening. And this is a mechanism that's been described in food, lotions, and oil and energy, and was described more than 100 years ago by actually a couple of scientists, but it's named for Spencer Pickering. and it's called a Pickering emulsion in food. And so you may have some of these items in your fridge. So ice cream, mayonnaise, all contain oil droplets that are stabilized by solid particles, or homogenized milk. And this is what got us really excited. So in homogenized milk, there's actually a protein, casein, that makes solid clusters that coat the surface of oil droplets. And you get an architecture that looks very similar to what we see in our MEG3 and PEGL3 emulsions. And so we wanted to know if MEG3 could prevent coarsening or prevent the droplet growth of PEGL3 droplets. And so I'm just going to really quick summarize some of this work to give you a little bit of an idea of what we found. So here we're looking at an in vitro condensate assay. We have PEGL granules, and we look at them over time. And so when you look at PEGL, these droplets get bigger over time. And we can quantify that here, looking at a histogram of radius. When we add the protein MEG3, now we see that well, hopefully you can see that the droplets don't really change over time. So they stay about the same size in the first image at 30 minutes as they are 3 hours later. And we can quantify this and see that there's really no significant change in size. So this suggests that at least in vitro, MEG3 can prevent coarsening. Okay, so we wanted to know then what would happen in vivo. And so to do this, we went back to our elegans system, and we have a really nice developmental time points where we can look at this. So MEG3 is really only robustly expressed in late oogenesis and in embryos, and not really expressed in the adult germline. And so it only begins to associate with granules in the maturing oocyte. So we thought that MEG3 might not have much of an effect on granules in oocytes, but we might see a strong effect in early embryos. And so we looked at this in wild type and MEG3 mutants. Okay, and so here's our starting system. Here's a wild type oocyte. You can see we have P granules that are mostly cytoplasmic and a one-cell embryo. Now, when we get rid of MEG3, in oocytes, we see very little change in the size of the droplets, but in our one-cell embryos, we now see fewer larger droplets. And we can quantify this. So if you look at the size distribution in oocytes, it's basically the same with and without MEG3. You see similar numbers and similar volume of condensates. But in embryos, we see something very different. So when you have MEG3, your droplet distribution shifts from a bigger, broader distribution of granule sizes to a much smaller granule size. And you see this big change in condensate number because you haven't had coarsening, but no change in the total number or the total amount of protein in condensates. All right, so this suggests that, yeah, MEG3 can form solid clusters on these PEGL liquid condensates, and that in embryos, MEG3 is actually important for regulating the size of these granules. Okay, so what exactly is MEG3? So unfortunately, MEG3 is a mostly intrinsically disordered protein. It has a C-terminal domain that is predicted to be structured, but is actually very poorly predicted. So if you're used to looking at alpha-fold structures, blue means that it's predicted with very high confidence, orange means very low. So we don't have a whole lot of structural information to go on, except that this is probably disordered, and this C-terminal domain has some structure. And so one other thing I should point out here is that I forgot to mention earlier, I just want to, you'll see it on some of my future slides. MEG3 actually has a duplicated protein. So it has a redundant paralog in embryos, which is MEG4. You can get rid of either one and embryos seem just fine. So we talk mostly about MEG3 because it's a little bit higher expressed. And in all the data I'm going to show you, we're working with MEG3. And in most of our in vivo data, MEG4 is deleted, so we don't have to worry about it. So yeah, I forgot to mention that. But MEG4 is almost identical at the sequence level. So it also doesn't give us a whole lot of clues to work on, even looking at how those paralogs diverge. So without much to go on, we just decided to chop it in half and take this intrinsically disordered domain, and the structure is C-terminal domain. And so if you follow the condensate literature, there's been a lot of attention on the role of intrinsically disordered domains and how they can perform, how they can assemble clusters or droplets in vitro. So we wanted to know if that was true in our system as well. Okay, so here's the in vitro data that I'm going to show you first. So here we're looking at PEGL in the absence of MEG3 and then with MEG3, and you can see MEG3 clusters and the droplets are slightly smaller. When we now look at our C-terminal protein or the IDR, we found, kind of to our surprise, the C-terminal domain actually still makes clusters. And the intrinsically disordered domain is very soluble. It's weakly recruited to the PEGL phase, but we don't see this surface localization or cluster formation anymore. And we can quantify that, in fact, these C-terminal clusters can prevent coarsening of the emulsion. So you have more smaller droplets when you have the full length or C-terminal domain, which you can see here in blue and green, whereas when there's no MEG3 or the IDR alone, you see larger granules, and they're almost identical to each other. So this suggests that at least in vitro, the C-terminal domain is what's making these solid clusters, whereas and not this intrinsically disordered domain. We can look at this in vivo too, and this is something that we're still really quantifying, but what we can see here, if you look at full-length MEG3, you have small droplets in a one-cell embryo versus large droplets in a MEG3 mutant. But if you look in the C-terminal mutant, you see a nice fine dispersion of granules, suggesting that the C-terminus is sufficient for this size control in early embryos. Okay, so that's kind of fun. Now we know MEG3 is important for the size regulation, and it probably requires the C-terminal domain. But the title of my talk is about mRNA regulation. And while I would like to tell you that having big droplets causes big problems for cells, we don't know. We can speculate on why you might want more smaller droplets versus large droplets. But as these embryos divide, these MEG3 mutants have other defects that perhaps become more important. And so in a wild type embryo, as it divides, you have a 2-cell embryo where you have condensates in the posterior and they've dissolved in the anterior. But in a MEG3 mutant, these granules don't polarize. And I'm not going to talk too much about the mechanisms of polarization, though that's something that we're also interested in. But what I'm going to focus on today is that we see that maternal mRNA is mislocalized in these embryos. And so that's what I'm going to focus the rest of my talk in, is what the role we think that MEG3 is playing in the localization of maternal RNA. Okay, right, so in embryos, this is a genetics department, so I'm hoping you are all familiar with this. The early steps of embryogenesis occur actually in the absence of transcription. And so almost everything is post-transcriptional regulation. So you have maternal RNAs and proteins that are deposited in the embryo. And in C. elegans, in the early cell embryo, what we see is that many of these maternal RNAs localize robustly with P granules. So here we're looking at a poly-A probe, which identifies the 3' ends of RNAs, and you can see that you get these bright condensates that co-localize with our P granule marker. There is RNA in the rest of the cell, it's just enriched in these granules. In a MEG3/4 mutant, what we see is that you completely lose this enrichment of RNA. So you still have P granules, they're now no longer localized, and they no longer enrich RNA. And so here we're looking at 4-cell embryos. We can follow this embryo a little bit further on in development to this 30-cell stage, which is the germline founder cell. And in wild type, you have RNA that enriches in P granule condensates. But in MEG3/4 mutants, that RNA enrichment is completely gone. So again, there's still RNA here. It's just no longer enriched over the RNA in the somatic cells. And we think this is actually probably pretty what may be really biologically important. And that's, we know that some of the RNAs, and I'll talk a little bit more about these later, that are in these germ granules are really important for establishing the germ cell program. And so in mutants that lack MEG3/4, what we see is that these germ cell founder cells fail to proliferate as you're going into the adult worm. And so 30 to 60% of C. elegans worms lack a germline in the adult worm, so they're sterile. Not 100%. So that means that the worms that do develop a germline are actually, as far as we can tell, nearly wild type in terms of fertility. And so we think that's because there's still RNA there, just not as much. And that I'll mention that this will come up again a little bit later as well. All right, so we had found that that MEG3 was important for RNA localization during my postdoc, and a fellow postdoc, Sean Lee, decided to perform iClip on MEG3. And so he pulled down MEG3 from embryos and looked at the RNAs that were cross-linked with it. And he found that there were about 500 RNAs that strongly bound to MEG3. And this is one of them here. This is a GRIF1 mRNA. And as you can see, it forms bright puncta. And these are, in fact, germ granules. And another graduate student in the lab, Alicia Scholl, went on to validate that almost all of the 500 of these RNAs are, in fact, germ granule mRNAs that enrich in germ granules. So in embryos, there are about 6,000 mRNAs that are expressed, that are maternally deposited, and only 500 of them are enriched in germ granules. We don't really know yet exactly what drives that specificity. We have a few clues. So if you look at RNAs that bind to MEG3, they're almost twofold longer than all transcripts in the genome. This is kind of interesting because this has actually been seen for many cytoplasmic RNA granules. So stress granules and P-bodies also contain RNAs that tend to be longer than than other transcripts. Additionally, MEG3 RNAs tend to be translationally repressed. So Sean also did ribosome footprinting in early embryos and compared the ribosome occupancy of all transcripts to those that were MEG3 bound, and found that on average, they have lower ribosome occupancy. So again, this is consistent with many cytoplasmic RNA condensates that seem to contain translationally repressed RNAs. But it's not, but we know that there are RNAs that are long and translationally repressed that aren't in P granules. So it's not sufficient to explain the specificity. But like I said, it gives us some clues. It also suggested that MEG3, that P granules might be involved in translational regulation. And so we wanted to look at this. So here we're looking at GRIF1. So this was one of the mRNAs that was a top MEG3 target. And we also know that it's translationally regulated during embryo development. So here on the top, you're looking at the RNA, which enriches into germ granules. Here we're looking at an earlier embryo stage, and then later, this is the P4 stage, or about a 30-cell embryo. It's protein translation, looking at expression of a GFP-tagged GRIF1 protein, is inhibited in early embryos, but then is activated in this P4 or 30-cell stage embryo. And you can see that really nicely. You have this really dramatic change in translation. And this is all maternally deposited RNA. So the regulation's at the RNA level. In our MEG3 mutants, there are a couple of things I want to point out. So this RNA in a MEG3/4 mutant is not in germ granules, so it's throughout the cytoplasm. But you see in early embryos, translation is still repressed, right? So you don't need to be in a granule to be translationally repressed. And then additionally, translation is activated at this 30-cell stage of development. The level of protein is low, which makes sense. You have a lot less RNA in this cell, but it is still activated, right? And so this suggests that MEG3 is really important for localizing the RNAs in germ granules, but not necessarily important for their translational regulation. And we've seen this for a few more target RNAs. Okay, so this suggests that MEG3 regulates granule size and RNA enrichment. And so we have this size effect in early embryos, but then as embryos start to mature, you have these really big differences in how maternal mRNAs are recruited to granules. So MEG3 does not have a canonical RNA binding domain. I showed you the alpha fold structure. N terminus is intrinsically disordered. The structured C terminus is who knows. But we've been working with Alex Holhouse, who works on intrinsically disordered proteins and is trying to develop methods to predict how they bind to RNA. And when he ran MEG3 through his predictor, what he found is that actually almost all of MEG3 is predicted to bind RNA. So here you're looking at the potential of a specific amino acid to have an attractive interaction with RNA, neutral in gray, or in green form a repulsive interaction. And what you can see is that with the exception of a few repulsive peaks in kind of the middle of the IDR, and then a few more in the C terminus, almost all of MEG3 is actually predicted to pretty favorably bind to RNA. And indeed, when we purify RNA, when we purify MEG3 and look at RNA binding in vitro, we can see that it can bind to a poly-U RNA in a gel shift assay. So here we're looking at titration of MEG3, and in fact, it binds to this poly-U RNA in a nanomolar range. So it's a pretty good RNA binding protein. And then if we look at this in our condensate assay, here we have purified MEG3, PEGL3. Now we're adding an mRNA, and this is NOS2, which is another MEG3 target. If we look at a close-up of a granule that doesn't have MEG3, you have a little bit of RNA. It's not completely excluded in your PEGL droplet, but when you add MEG3, you now have RNA that's enriched in these MEG3 clusters on the surface of the granule. So this suggests that MEG3 can bind RNA intrinsically, and that it can also recruit it to a condensate. Okay, so Jian Li, who's a graduate student in my lab here at UW-Madison, has been trying to understand how this protein interacts with RNA in more detail. And so to start, we just started by chopping up the IDR. Again, we don't have a whole lot of good clues for exactly where the most important regions will be. So we thought we would just kind of do the brute force attempt first. So she made these mutants by using CRISPR to make the mutants. Each of these proteins has an OLAS epitope tag, which lets us look at the protein expression and localization. And then she used these mutants to look at RNA localization. So, okay, so there's going to be a lot of images on the slide. I wanted to show you all these mutants together. So I'll walk through this quickly. So again, we have a wild type embryo. So this is a 4-cell embryo. We have mRNA and P granules that are localized in the posterior, and they co-localize together. In our MEG3/4 mutant, you lose that RNA enrichment. P granules are still there, but they're throughout the cell, and they don't co-localize with RNA. If we add back the C terminus, so looking at PEGL first, you have granules. They actually partially polarize, which is kind of a topic for another day, but they aren't completely polarized. And when we look at the RNA, they still have very low levels of RNA. As we add back 150 amino acids of the IDR, we actually start to see RNA that's enriched in germ granules, not as robustly as wild type. And they co-localize with our P granule marker. And then as you add back another 200 amino acids, this worm actually has nearly wild type levels of RNA co-localization, and even though it's lacking the first 200 amino acids. And we can quantify that, which is shown here, which is just looking at the volume of RNA in this P cell. And so, yeah, in MEG3 mutants, you have much less RNA in the germ cells. And then as the IDR length increases, you have an increase in RNA. And this mutant lacking the first 200 amino acids really looks very similar to wild type. And we can also perform these experiments using single molecule FISH to one of our MEG3 specific RNAs. So Jian did this looking at GRIF1. And we can see that this follows a very similar trend. So the data here looks almost identical to what we see with poly-A RNA, where as you increase the length of the IDR, you see kind of this gradiated enrichment of RNA. All right, so what we really would like to get at is this biologically meaningful? And so Jian then looked at the sterility of these worms. So again, in MEG3 mutants, the germline cells fail to proliferate. This gives us a very easy phenotype to quantify. So in a wild type worm, most of the worm is actually germline. And so you can see this just under a bright field microscope. But in these MEG3 mutants, they just kind of an empty cavity that shows up as a clear or white cavity in the worm. And so you can easily quantify the percentage of sterile worms. So in wild type worms, they're completely fertile. You have no sterile worms. In a MEG3 mutant, it's about 60%. And then as we add the C terminus and then increase the length of the IDR, what we see is that you have a really nice correlation of sterility with the RNA enrichment that we saw. And so this suggests that this correlation with RNA may actually be very important for the biological function of MEG3. All right. Okay, so I want to spend a little time on another biological phenotype of MEG3 mutants. So this is a phenotype that you actually can't see just looking at the worms. It's more a stimulated phenotype. And so, and I'm sorry, I'm not sure if there's a way I can get the headers to show up on the top, but what we see is that MEG3 is also required for exogenous RNAi. So if you give worms double-stranded RNA, you can either have this in their food or you can inject it into them. You can knock down genes, right? So this is just exogenous RNAi responses. But in MEG3 mutants, they're unable to do exogenous RNAi. So you can give them double-stranded RNA and they are completely insensitive to it. And so there's a lot of work that's been studied on this phenotype. It's not only MEG3 worms that have this phenotype, but also just worm strains that have disrupted P granules in embryos have this phenotype where you can't do exogenous RNAi. And so some beautiful work from the Seydoux lab, Kennedy lab, and the Rechevi lab have kind of worked out some of the mechanisms. And I'm going to give you a really brief overview of what's going on so you can kind of understand the phenotype we're looking at. But I'm happy to talk about this more anytime. Okay, so here's kind of a stereotypical cell. You're looking later in embryogenesis around the P4 stage. You have your nucleus here and a P granule that's associated with the nucleus, and then your cytoplasm in yellow. And so what we know is that actually C. elegans have a lot of endogenous RNA pathways. So they make small RNAs. And these small RNAs actually target genes in C. elegans. And in fact, most RNAs have a small RNA that targets them. But in the worm, it has a way to choose which transcripts are actually targeted or not. And it's believed that this has something to do with P granules. And what you see is that when you disrupt P granules, RNAs that were previously protected from silencing are now targeted by small RNAs. And in MEG3 mutants, this is what we think happens. Because those granules don't polarize, you have a lot fewer P granules in later embryos, and you get a very similar phenotype to mutations that disrupt germ granules. Okay, so this is the endogenous small RNA targeting that I was talking about, but it's exogenous RNAi that MEG3 worms are actually not able to do. And this is because a subset of the RNAs that are targeted for silencing in these mutants are actually required for exogenous RNAi. And so one of those genes is SID1. So this is actually a double-stranded RNA importer. So it's required to import double-stranded RNA into cells. And so in the absence, when this gene is not expressed because it's now been silenced in MEG3 mutants, the worms can't even take up the double-stranded RNA to elicit the phenotype. Okay, so there's a lot of cool, complicated biology in here, but the important part is that MEG3 worms can't do RNAi. And so there's some really nice assays that we can do to measure this. So the experiment that we're doing here is we can feed worms this POS1 RNAi. This is required for normal embryo development. So when you target this gene, embryos don't hatch. But if your RNAi doesn't work, then you get normal, all of your embryos should hatch. So if we look at a wild type worm, which is shown here in black, and on a control RNAi, so you're not targeting POS1, you have nearly 100% viable progeny. But when you use the POS1 RNAi, all of the embryos fail to hatch. In a MEG3 mutant, however, they're resistant to this silencing of POS1, and you have 100% of the embryos hatch. So now when we look at our mutants, all of them respond the same on the control RNA, so we see that progeny are viable as we expect. But then as we look at the viable progeny, we see in the C-terminal mutant, which had almost no RNA recruitment into the germ cell, you see that these worms are like the MEG3/4 mutant, completely resistant to RNA. But in our MEG3 truncations that can start to recruit RNA into the germ cell, they are now sensitive to RNAi. The 200 to 862 variant of MEG3 is almost completely sensitive, very similar to wild type. And this 401 to 862 is maybe partial sensitive. So we're still trying to characterize this, this phenotype, but you get this kind of intermediate phenotype between the C-terminus and this mutant. So again, this suggests that this recruitment of RNA into the germ cell and potentially, polarization of P granules is important also for these RNAi defects. Okay. All right. So now we've added, MEG3 is important for size control in the early embryo and RNA enrichment. And this requires the intrinsically disordered domain, but it's not just an all or none. There's maybe not one binding site. We instead see that you get partial recruitment when we lose part of the IDR, as opposed to an all or nothing phenotype. And so we wanted to understand a little bit more how MEG3 binds to RNA. And so we looked at MEG3 RNA binding in vitro. We purified a whole bunch of different MEG3 truncation constructs that either lack portions of the IDR with the C-terminal domain, or just have the IDR with truncations in the other direction. And what we see, and this is Joey Larson in the lab, purified all these MEG3 mutants. And so what we see is that, as we had previously found, full-length MEG3 binds RNA really well. So this is shown here in black. You get an apparent affinity of about 100 nanomolar. As you lose the first 200 amino acids, you see a decrease in RNA binding, but it's still pretty good. So you're about 380 nanomolar. And this is the construct that we saw was nearly wild type in worms. When we lose the first 400 amino acids, our RNA binding goes almost to zero. We think it still does have some RNA binding activity, but at least to the short poly A, it's very weak. On the converse, when we look at the IDR, we see that the IDR binds with, again, about 100 nanomolar affinity. So the IDR is really what's driving RNA binding. We really don't see much effect of the C-terminal domain. And as you truncate this, so this region from 1 to 400 binds RNA with high affinity, and this region from 1 to 200, which wasn't required in vivo, actually still binds RNA really well. So this suggests that you can bind RNA along potentially multiple regions of this IDR, and we're still working out to define this more clearly. Okay, so let me see. All right, yeah, I should have time. Okay. So the last little bit that I want to share with you is how we're thinking about these RNAs and condensates. And so I told you that MEG3 makes solid clusters, and we can see that here. So if you take a MEG3 little solid cluster and photo bleach the fluorescence, it doesn't recover over time. So these are solid particles. They're not dynamic. If we now look at RNAs, however, well, we can see if we use a short RNA, so this is 30 nucleotide substrate, similar to what we used in our binding assays, This can get recruited into a MEG3 condensate, but it's very dynamic. So when you photobleach it, you get recovery. So even though you have a solid MEG3 condensate, these short RNAs can be dynamic. But as you increase the length of the RNA from 100 or to a full-length NOS2 RNA, you see that these dynamics really slow down. So with 100 nucleotides, you see recovery, but it's low. Once you get to 200 nucleotides or to a 1200 nucleotide mRNA, you see almost no recovery in these MEG3 clusters. And so this suggests that MEG3 can actually stably bind long RNAs in a condensate. And so we can actually, so we wanted to know if this was true in vivo as well. And so we don't have very good tools for looking at dynamics of RNA in cells. But we have an assay that I think is pretty cool that we can do in embryos. So embryos have an eggshell. We can wash them and resuspend them in whatever buffer we want. And then we can poke a hole in the eggshell, and this causes the cytoplasm to extrude. We're essentially diluting the cytoplasm. And so for a liquid droplet, you expect when you dilute it, it will dissolve. For a solid droplet, it's going to stay as a condensate. And so if we do this experiment, so I just shine a laser, this embryo exploded, Peggle dissolves instantly, it's a liquid droplet, but you have these stable MEG3 clusters. And so we can then use this to look at RNA. So we use cyto14, which is an RNA dye. We can soak it with permeabilized embryos, and it gets taken up in the embryos. And we can see that it forms puncta. So we are hoping that it binds to RNA, and they co-localize with MEG3. We can form the same extrusion experiment. And when now we have MEG3 puncta that are stable outside of the embryo, and we see that the RNA is still enriched. And we can quantify this here, and we see that really the RNA interacting with MEG3 seems to be stable ex vivo, suggesting MEG3 can bind long RNA stably in these solid clusters. And that may be important for how it's enriching RNAs in cell, where you're stably binding RNAs. Okay, and so again, we think MEG3 is important for size control, for RNA enrichment, and that it may work by stably binding RNAs. And I think I will just a little summary model here. So we're thinking about, how this may factor for other proteins. So MEG3 is not very well conserved, but it contains this long IDRs, and there are a lot of proteins with long IDRs. And so we're kind of thinking of a model where this IDR can bind RNA in many places. So if you have a short RNA, you could get multiple RNA binding sites. As this decreases, you have fewer binding sites, but you can still bind RNA. With a long RNA, this is going to bind to multiple places on the IDR, and this is going to give you high affinity through avidity. You can make multiple interactions. It's going to be stable because it's hard for this whole molecule to dissociate. But as the IDR gets shorter and you make less contacts, this may change. And so we're interested in seeing how these shorter IDR constructs, if that will change the dynamics of RNA and granules. And also thinking about this, when we're thinking about what are the functions of IDRs, it may not only be sequence that is important for what they're doing, but really how long they are. And so with that, thank you for your attention. And I'm happy to take that. Yeah.
So my research interests primarily revolve around space biology and the physiological challenges that are associated with long-term spaceflight. So obviously surface tension becomes a dominant force in the absence of gravity. So I'm wondering if you have any hypotheses regarding how this phenomenon might affect the formation of granules and by extension embryonic development in spaceflight.
Yeah, so C. elegans have actually been sent to space, and I'm pretty sure they developed at least somewhat normally earlier, but I don't know, it'd be interesting to think about. I haven't thought about that much. So that's it'd be really fun to look at what happens to P granules. Yeah. Thanks. Yeah.
So I have two questions. First, apart from the role of Pegasus in RNA-mediated mRNA silencing, does it play any other role in the recruitment of mRNAs to the MEG3 protein? like if the MEC3 is not on the PEGL3 droplet by some experimental perturbation, does that affect the mRNA recruitment to the MEC3? And second question is in the context where the PEGL3 droplets are larger in size in oocyte as opposed to the embryo. In that case, is there a specific limit to the size of the PEGL3 droplets in the oocyte? And what, if so, what would be limiting the size of the droplets in the oocyte?
Yeah, okay, great. So the first question was, about RNA recruitment. So we can actually get rid of PEGL3, PEGL1 proteins in the embryo. You get smaller mEG3 granules, but they still have RNA. There are other factors though. So I would say that's something that we still don't fully know is, how MEG3 can do it independently, but we know that you can get rid of at least the PEGL proteins. For the second question, okay, yeah, so granules, they're bigger, right, in early embryos. And so here we, it's probably time is an element that limits granule size because these embryos are going to start to divide. And as you get larger, fewer granules, so granule growth that what we see in embryos is actually mostly through droplets colliding with each other. As you get bigger, fewer granules, the chance that they're going to collide happens a lot less often. And the embryos just divide before that happens. I mean, and we can do some things though where you can potentially arrest that. And we don't see that granules get bigger. And we think that's also because there's some regulation of granule dynamics by kinases that are cell cycle regulated, which is kind of fun and also play a role in how these granules are polarized. But yeah, great question. Yeah.
What happens to the contents of the granule when it dissolves? So when you have that polarization and the dissolution, the RNAs and proteins get degraded, or do they end up becoming incorporated into the granules at the whole?
Yeah, no, it's a great question. So there's so essentially for what we can mostly tell, they dissolve, so they become soluble. So we've done this not for every protein, so I can't tell you that it's true for all of them, but the concentrations in the cell don't change, right? It's really just you have a droplet and they're dissolving. And we do think that's regulated by post-translational modifications that actually change the solubility of the protein. But then in the embryo, there are also proteins that get localized in cytoplasmic gradients. And so some of these proteins, they're getting localized in a cytoplasmic gradient at the same time. So they get enriched in the posterior half where you see these kind of bigger germ granules.
They're showing up.
Yeah. Yeah.
Thank you for the class. I have many questions, but I'll ask you just one that is related to the previous question about how much the numbers or intensity of these granules affect the hatchings or the potential fertility of these adults?
Yeah, so that's a great question and something that we could look into more. And one of the ways that we maybe looked at this just a little bit is the size of these granules is temperature dependent. And so this is one of the, this is, I think, something that we should look at, and we could look at. Definitely up to a certain temperature, you can have smaller granules and they're still fine. And this may be in part, you can see here, you can lose quite a bit of, for example, RNA binding in smaller granules, and worms still seem fairly fertile. But there is a point that you cross, which is actually at about above 25 degrees Celsius, where you start to see fertility defects, especially in germ granule mutants. And as you increase the temperature, that is exacerbated. So but I'd say there are a lot of things happening. So it's not really clear if it's granule size or if there are other things that are going on. I mean, I guess what we see in our mutants, right, is even in a MEG3/4 mutant, they're not 100% sterile, right? They're only 30 to 60% sterile. So the system is actually pretty robust, right? And you don't need 100% granules. You barely need RNA enrichment. You don't even really need RNA enrichment at all to get at least a fraction of your embryos that develop normally. But I think, is there a threshold where you start to see sterile worms, and does that correlate with granule size? I mean, it's a great question. Yeah. Okay, anything else? Oh, yeah.
The worms, the mutant worms that are fertile, have you ever select for them? Like something make them stay fertile even when they shouldn't be? Do they have other stuff that, because I guess you're using a population that would be not like isogenic.
Yeah, so I'll answer this, maybe just going back really quick to this slide. Right. So the question is, what's happening in the worms that are fertile? And so what our model is right now, and maybe hopefully we can test this, and this is a reporter that I want to look at, right, is that so in worms that don't have MEG3, the RNA is not gone, it's at lower levels, and you're still making protein. And so our hypothesis is actually, so for example, GRIF1 is required for the turnover of maternal proteins, and is required to inactivate for your normal germ cell program to take place. But it's actually, it's redundant with other proteins. Again, there's a lot of redundancy. So you only see this phenotype in combination with loss of other maternal mRNAs. And so it's possible, and what we think is that, you're making some protein here, and it may be just stochastic. In some embryos, they get just enough protein to get them over the threshold. And if you do that, and if you start to initiate the germ cell program, then it just takes off, and the worms you get they're normal. And so it's a threshold effect. And so in these worms where we're seeing that you get increasing amounts of RNA, you're more likely to get worms that are going to cross that threshold. And that's why we see a decrease in sterility. So that's our model. And part of that is because, so for example, there's variability between worms. So if you may have a worm that has, whose offspring are 80% sterile, and one that is 30%, their offspring are 30% sterile. But if you take the fertile progeny from that generation, you get the same sort of kind of random distribution. And so we think, yeah, there's at least some component in there that's kind of stochastic. Yeah, great. Yeah.
Just wondering if you wanted to look at the embryo, like an embryo born from a heterozygous like hermaphrodite versus like a homozygous mutant for MEG3, and if there's any differences between them?
Yeah, so we have done this. Well, not exactly to look at it. I mean, so it's a maternally deposited protein. So I'm trying to think. We haven't done exactly that experiment. We tried looking at differences between MEG3 and MEG3/4, and whether you have MEG4 present or not. And there do seem to be some slight differences in the condensate size, but I don't know that we've actually ever done that with a heterozygous MEG3. That'd be something we could do, though.
Yeah, 'cause that would technically like the mutant phenotype again be well based on the model since it's maternally deposited, right? Like the heterozygous would deposit it to the embryo, and because it's a mutant, they wouldn't have that phenotype.
Yeah. Yeah. Oh, though, no, I just, yeah. Yeah, no, yeah, right, that's possible. I'm thinking about the details, why we haven't actually done that, or if we have. So, but I don't think we have, but we could, yeah. Yeah. Great. Okay, thank you so much.